So the next section of the book is going into the different types of white blood cells. Again, that's straightforward memorization. There's really no tips or tricks that I can tell you. I can go through a little bit of how to diagnose them under the microscope. I'm not sure if your lab instructor is going to have you do that or not. One of the first things you need to use as a diagnostic is whether or not the white blood cell is grainy or not. If it appears grainy, if that nucleus looks like it's blotchy in different parts, then it's not a lymphocyte or monocyte. Lymphocytes and monocytes have nice smooth nuclei. Monocyte nuclei kind of look like a kidney bean or almost a C shape or a boppy pillow, if you know what those are. That's what a monocyte looks like. They're very big. Lymphocytes are very, very small, and their nucleus is smooth and takes up almost the entire cell. Basophils and Eosinophils look very, very dark and spotted under the microscope. It's almost impossible to tell them apart. And neutrophils are the most common. If you see a white blood cell under the microscope, it's probably a neutrophil, especially if the nucleus looks very splotchy or if it almost looks like it has 4 or 5 nuclei in it. It's probably a neutrophil. Anyways, moving on from that, you will also be able to identify platelets under the microscope. They're little pink flecks, little pink spots under there, kind of sprinkled in, and sometimes they're clumped together, but usually they're just kind of sporadic in your field of view. Hemostasis is the next section, and that's the fancy word for blood clotting. It's important to understand how the blood clots and the systems involved. It starts with something called a vascular spasm, and during that vascular spasm you have the different layers of the blood vessel constricting back to basically allow blood to flow through. And what that leads to is a platelet plug formation. So as the blood's flowing through, all those little bits of platelets that have been floating in the blood start to stick. They start to stick to those open ends of the blood vessel, and as that happens over and over and over again, a small temporary plug will form. It's called a platelet plug. So while that's happening, there's all of these signals that are going on in between the cells, and they're called interleukins or prostaglandins. They're basically little cell-to-cell messengers so that the cells in this broken area can communicate to the cells in the non-broken area. Hey, we're broken over here, just letting you know. We might have some invaders coming in. We might have to increase blood flow or start this inflammatory response. So it's just to let the other cells know what's going on. This whole cascade pathway takes place, which is way above AMP2 level. But happens. Then coagulation occurs. Coagulation is the actual clot formation. So the platelet plug is temporary. It's just a bunch of platelets kind of stuck on top of one another, temporarily sealing that patch. But it's not going to hold for very long. Blood pressure is relatively high. It's rushing underneath. And so there needs to be a more permanent fixture, and that's what the clot is. The clot is made from structures called fibrin. Fibrin is exactly what it sounds like. They're fibers. And there's this whole cascade pathway that takes place in order for fibrin to form. And you see that pathway on page 653. There's a whole lot of words in there and a whole lot of cofactors and vocabulary. What do you need to take away from this chart? Well, there's two things. Let me get my handy dandy whiteboard here. Let's see how I can prop myself up. There are two pathways involved in blood clotting. One is extrinsic. Do you see that? Kind of. Extrinsic means it's on the outside of the blood vessel. There's also intrinsic. That's on the inside. So in other words, when this blood vessel, let's see if I can do this. Do it like that. When this blood vessel is broken, right, this is the wall of the blood vessel, this is the wall of the blood vessel, blood flowing kind of through here, and we have this break here. This is the wall, this is the wall, right? The blood vessel repair is going to take place on the inside, intrinsic, and the outside, extrinsic. All right, does that make sense? I know it's kind of small. My handwriting's terrible. So that platelet plug forms, and again, that's just a temporary little bubble that occurs. As that there, kind of stabilizing, this whole cascade pathway that you see on 653 is taking place. And it all starts with the activation of what's called factor X. Factor X is necessary for clot formation to take place. There are some other things that need to kind of go forth from this. So again, intrinsic and extrinsic pathways are both taking place to fill in this whole area. Right after the platelet plug is formed, that whole area will fill up with the extrinsic and extrinsic pathways that you see here on page 653. Factor X is the beginning of what's called the common pathway. In order for factor X to be activated, vitamin K, I'm just making sure your book even tells you this, vitamin K is needed. It doesn't really tell you this, but vitamin K is needed, and calcium is needed also. So you need calcium and vitamin K in order for that common pathway to start. And the common pathway is simply a cascade chain reaction that takes place. Once factor X is activated, then prothrombinase converts prothrombin into thrombin, and then thrombin converts fibrinogen into fibrin, and fibrin is our clot. So once factor X, let's say factor X has to be activated, and that requires vitamin K and calcium, Ca. Oops, sorry about that. That is going to lead to that prothrombinase turning prothrombin into thrombin, fibrinogen into fibrin. Boom, there's our clot. Think about the scab that's forming, or think about internal damage if you have a bruise. The same pathway is happening. Hopefully this video confusion will help you. I'm going to get some more colors because these kind of stunk a little bit. Sorry about that. So again, what I would do, you don't have to know all those factors that are listed on page 652. Factor X is the only one you need for my class. There's this whole crazy pathway. I would paraphrase it. Again, leave out some of that extra stuff that I didn't go over. Extrinsic and intrinsic lead to factor X. Vitamin K and calcium are important. And then that whole common pathway of prothrombin to thrombin, fibrinogen to fibrin. Important. All right? Then the last little piece of hemostasis is, of course, the clot retraction. Once the scar tissue is forming underneath, the epithelium and endothelium are replaced and renewed, and scar tissue and collagen have built up, then eventually that scab can dissolve and fall off. So that's basically what happens. Of course, it's not that easy. There's this whole pathway, and there's some enzymes involved. Those are listed there on page 654. Again, that's memorization. So we'll stop here for this section of the video.